Yeah. So, welcome guys. I hope you can listen me. Yes, boys. Kalyan, Sai Kumar, Asya, Arti. Yeah, we are
And
going to start aseptic processing. What is
So,
aseptic processing? I already said you. A means without. Septic means without a sepsis, without an infection. So a processing in which you don't give any chance for the bacterial contamination or bacterial growth or any sort of infectious pathogen, bacterial viral growth, right, is basically called as aseptic processing, right? So aseptic processing is very important in the field of preparation of parenterals, especially these knowledges which we gain from the microbiology, right? So this is very important in preparation of parenterals, because in other formulations we are giving, like for example tablets or capsules or syrups, we are giving it orally. So they basically go into the stomach. You have certain lag time before they reach to the blood. So you have a lot of other systems, right? You have first line immune system to act on it, right, if any bacteria or pathogen is present. But in parenterals you're directly giving it into the blood, or you're instilling into the eye, or you're directly giving it to your nasal tract, right? So there is immediate chance that they are directly going into the blood. So there is chance that they can infect you. That's why we must be pretty sure that there is no chance of infection while preparing. That's why we will follow aseptic processing, right? So you can see clearly how he is handling with that clear picture. It's showing the way he dressed, right? So in aseptic processing we maintain basically three different things. We maintain the material entering into the room, we maintain the person entering into the room, and we will maintain the quality of the air entering into the room. So everything which is entering in this room should be free from particulate matter, dust, bacteria, pathogens, and everything. So we should carefully monitor. You can see a person entering into that room. He is fully covered. He has a headgear. He has a mask. He has the right, everything. Gloves are there from top to bottom, from toe to his head. Everything is covered, right, systematically. And this is how the people enter into the aseptic area, because whatever bioburden should be decreased in those areas. Anything present on your hair shouldn't fall down there, right? Or anything present in your body, or the dust or the dirt or any pathogen present on your fabrics, on your clothes, shouldn't affect the aseptic environment. That's why all that setup. Then material also enters carefully. There will be exchange boxes. I'll show you. Material will be placed there, then an air will be blown on it, then UV radiations will be blown on it. Then it was make sure that no bacteria is present. If at all it will die, then they will take inside. And with interlocking doors, right? So there will be a cabin, interlocking doors. So any material to which should come in a septic room should be placed in that cabin. Door is locked. When the outside door is locked, then inside door can be opened. That's called interlocking door mechanism, right? Then again they will sterilize it. Then inside door is opened after locking of outside door, and they'll remove it out. Even persons entering into the room, there are exchange rooms. So there is no direct door between two rooms in pharmaceutical companies, especially in aseptic areas and in areas where parenterals are prepared. There are exchange rooms between two rooms, like we have one in our lab, right? If you go to the formulation technology lab, so between two labs there is a preparation room. So that acts as an exchange room and two interlocking doors. So first you will open and you will enter into that room, then you will change your clothes, right? You will wear all these protective these things, right? Then you will be blown with hot air, then you will sanitize yourself. Then after locking of that door, preparation room, next door will open. So these exchange rooms are also there to maintain the quality, right? And air entering into the room is also monitored. So how air entering in the room can be monitored? Through HEPA filters and laminar air flows, right? Even the quality of the air, particulate matter present in the air, dust particles, even the flow of the air. So without turbulent flow, అది smooth గా laminar flow వచ్చేటట్టు you use these HEPA filters and laminar air flows. And we even use dehumidifiers and other kind of things in those particular rooms, right? So we will study in coming class also that there are different classifications like gray area, black area, white area. the class 100 room, class 1000 room, class 10,000 room, all these things we will study in aseptic processing, right? So asepsis means a state of control, right, attained by using a aseptic work area and performing activities in a manner that precludes, it means excludes, it means removes microbiological contaminations of the exposed sterile product. So this processing is called as asepsis, a state of control attained by using a what aseptic work area and performing activities in a manner that precludes microbiological contaminations of the exposed sterile products, right? So asepsis is also a practice to reduce or to eliminate contaminants such as bacteria, virus, fungi, and parasites from entering the operative field in surgery or medicines to prevent infections. Ideally, a field is sterile, free of contaminants, a situation that is difficult to attain. However, the goal is to eliminate all the infectious substances, right, which are not sterile. Okay, so this is the basic definitions of asepsis. So there should be no chance for the parasite, bacteria, virus, fungi, anything, maybe in the surgical area or maybe the medicinal preparation area, the no chance which can further lead to the infections. So practically it's very difficult to attain such kind of situation, and attaining that kind of situation in an area is called as aseptic area, and that process is called as aseptic processing. Okay, and whatever process you do in this is basically called as aseptic processing, preparations like formulations or any other things. So aseptic processing is processing of the drug components, drug products, containers, excipients, etc. in a manner that precludes biological, microbiological contaminations of the final sealed product is generally called as what? Aseptic processing. Sterile drug manufacturers should have keen awareness, right, of the public health implications of distribution, distributing a non-sterile product, right. So if I'm a manufacturer, I should know that what will happen if I distribute, if I release in the market and non-sterile product, right? What can happen? I should be aware about that thing, right? And poor CGMP practices. I hope you know what is CGMP, good manufacturing practice conditions. So GMP should be followed. We will have another class on GMP, good manufacturing practice. What are the different techniques and what are the main importance of, right, documented good manufacturing practice? We will see that. So poor good manufacturing practice, if they follow, then what will be the implications? They should know it. At a manufacturing facility can ultimately pose a life threatening health risk to the patient. So what will be the implication? It will be life threatening and health risk to the patient. So we should be aware while preparing the sterile drug manufacturing, right? So we should know what will happen there. So you can see sepsis, right? So sepsis, what is sepsis? You can clearly see there. Sepsis is a serious medical condition characterized by a whole body inflammatory state caused by infection. You can clearly say, you see in the picture how and what the sepsis looks like. So what will be the progressive symptoms of the sepsis? There will be a mild fever, decreased blood pressure, rapid breathing and heart rate, and skin lesions on the skin. You can see the picture there. Spontaneous blood clotting, organ failure, and in some cases, it can also lead to the death. So sepsis, septic avadam, we commonly say, right? So we commonly say if you have any infection, if you have any wound, we always say, see, take the tetanus toxide TT injection. If not, you will have the sepsis, right? So what is sepsis? You can see clearly. Sepsis means in those symptoms, it can lead to the death, till starting from fever, then decreased blood pressure, then rapid increased heart rate and the breathing, then lesions on the skin, then spontaneous blood clotting, organ failure, which leads to death. So you should be cautious about the infections, especially sepsis. It's dangerous. Sepsis means it's not a normal thing. Okay, it can lead to the death also, and these symptoms should be carefully monitored. Okay, right. So lack of sterility issues, you can see number of recalls. Recalls means basically the substances or the drugs products which are already distributed in the market are recalled. They recalled back, right, depending on their sterility issue. So you can see how many are recalled, right, in years till 2002. You can see here, right, that in millions how many products got recalled. So in the year 94 and I hope in 95 the recalling is less, but when you compare it from the year 98 to 2000, then till 2002 also sterility maintained చేయకపోవడం వల్ల many products are being recalled. So lack of sterility is the first reason for the drug recall in the last 5 years. So if you see last 5 years, so what is the reason for recalling of the drug back to the manufacturing area? Lack of sterility. So imagine how much economical impact will be there on the on the pharmaceutical company. See, they generally to prepare a new drug, to formulate a new drug, right, which is not existing in the market, it will cost more than 2 3 billion dollars, right. It will be generally equal to like releasing one new rocket in the space. So they're spending all that amount in manufacturing the things, in right, in marketing, and it's sending into the market. And when suddenly much of the product, or like 40 50% of the product comes back, so it is really an economical burden. So any pharmaceutical company doesn't want this thing to happen, right? They always want their products to be sold well advanced, well before its expiry date, right? But if it's coming back, it's not good for them. Generally, there are many formulations, at least 5 to 10% formulations will be coming back because of expiry dates, because of damages, or because of some other kind of things, ADRs, maybe other kind of things. But the major reason in last 5 years of the recalling of the drug back to the manufacturing industry is lack of sterility. So imagine how much economic burden it will keep, it will pose on the pharmaceutical companies. So nearly all drugs recalled due to the lack of sterility assurance in last 20 years were produced via aseptic processing, right? So last 20 years లో కంపేర్ చేస్తే aseptic processing లో చేసిన చాలా వరకు డ్రగ్స్ ని రీకాల్ చేయాల్సిన అవసరం పడలేదు. సో aseptic processing, if they don't follow aseptic processing, then there will be chances that you should recall the what drug due to the lack of sterility issues. Now, so producing sterile drug products, there are two basic ways of producing sterile drug products. You can see here, like we also discussed in yesterday, we said about terminal sterilization, we discussed about terminal sterilization, right? So aseptic processing is there, terminal sterilization is there. Two are different terms. Even these two are also sterile procedures only, but there is slight variation in aseptic processing and in terminal sterilization, right? So let's see. In terminal sterilization, the word itself says end point, terminal stages low, we will go for sterilization. So product containers are filled and sealed under high quality environmental conditions, right? Then designed to minimize contamination but not guarantee the sterility. Whereas aseptic processing, each and every step was went through aseptic way. So in aseptic processing, drug product containers and closures are right are subjects to sterilization separately and then brought together. We have seen in small volume parental preparations, right? Schematic preparation. I shown you. First, we took water for injection. We heated it until 120 degrees centigrade for 20 minutes. Then we subdivided it into two parts. One part is kept aside. The other part was right taken, and the temperatures decreased to less than 60%. Then we added preservatives and other excipients. Again, the temperature of that water for injection with preservative and other excipients was reduced to 20%, right? 25 to 30%, that is brought back to the room temperature. Then the drug is dissolved, right? Everything, every step is going through aseptic way. Clear? So drug products, containers and closures all are subject to sterilization. In terminal sterilization, everything was mixed. Ultimately, at last stage before labeling or after labeling, they'll send it for the sterilization. But here, containers are separately sterilized and brought back. Aseptic processing. Closures separately sterilized and brought back. Raw material are sterilized and mixed in aseptic conditions. Got it? So there is less chances of contamination, right? So next you can see terminal sterilization, product in its final containers is subjected to sterilization, and such sterilization processes such as heat sterilization or radiation sterilization. So in radiation sterilization, we use which radiations? UV radiations, right? Heat sterilization, we can use either moist heat sterilization using autoclave, depending on the drug product, or dry heat sterilization. So in hot air ovens, we use dry heat sterilization. So after filling them in the containers, sealing them, then we are going for sterilization. That is terminal sterilization. Whereas in aseptic processing, each and every drug product, excipients, drug, containers, closures, everything is sterilized, then used. So because there is no process to sterilize the product in its final character container, it is critical that containers be filled and sealed in extremely high quality environment. So second thing here, as final product is not being sterilized here, everything, filling, sealing, right, uh, packing, labeling, everything should be done in high quality environment. What does high quality environment means? High quality environment means, I said you, we will maintain three things: air entering into the room, person entering into the room, material entering into the room. Three things should be carefully controlled and monitored. So in such kind of environment, you are making the preparing the aseptic processing. Okay, so that's the basic difference between terminal sterilization and aseptic processing, right? So you can see here, terminal sterilization process must be compatible with all components. Terminal sterilization, drug product is taken, mixed, right? Then filled in containers, excipients also taken, added in containers, mixed properly, sterilization process, sterile drug product was got. So after mixing all those things, you are going for sterilization. Okay, whereas in aseptic processing, you can see drug product. Yeah, first drug product తీసుకున్నాం, sterilization process is carried out, sterile drug product came, containers, sterilization process is carried on, sterile drug containers I achieved, then closures, sterile process, sterile closures I got, excipients underwent to sterilization process, sterile excipients I got. Then I'll take all those things, I'll go for aseptic processing, I'll, right, fill them in, sterile final product is achieved. So what is the difference in these two, in old one and new one? In old method, that is terminal sterilization method, you will mix everything and the final containers, packed and sealed containers of sterilized terminal stages, end stages. Whereas here, every step there is a sterilization, right? So that is the basic difference. So when, if you see the global scene, European agencies for the evaluation of medical products, EMEA, terminal sterilization, right? So how much time it's taking? More than 15 minutes, right? And adjunct processing, more than 8 minutes is happening, right? So then aseptic processing, and in aseptic processing, uh, it's happening later, right? Then you can see what is PNSU, probability of non-sterile units. So here, probability of non-sterile units is more than 1%, right? So where, when terminal sterilization is happening, so the probability of units, product containers being non-sterile after the application of the lethal agents, PNSU of 1 in 10 to the power of 6, right? 10 to the power of 6 is how 1 million, right? So the probability that a unit is non-sterile is 1 in a million. Eppudu terminal sterilization చేసినప్పుడు, right? Then F4 sterilization process equivalent time. So the equivalent number of minutes at 121.1 degrees centigrade delivered to a unit by sterilization process. So probability of non-sterile units in aseptic processing, it is impossible to scientifically determine the probability of non-sterile units. See, this is the difference between the terminal sterilization and aseptic processing. In terminal sterilization, there is a probability of 1 in 1 million of PNSUs, and non probability of non-sterile unit, existing of a non-sterile unit, maybe one bacterial strain, one fungal strain, right? Maybe in million units. Okay, so million containers lonchi okka container lo there can be a probability of non-sterile unit presence. But in aseptic processing, it's difficult to measure the availability of probability of non-sterile units. So which was better, aseptic processing or terminal sterilization? aseptic processing, right? So many critical systems are involved, like processing rooms, equipments, personals, and everything in aseptic processing. Contamination rate, access with the media fill, right? So a simulated production run with media that promotes growth of bacterial organisms. PSNUs, probability of non-sterile, probability of units being a non-sterile after the application of lethal agents. PNSUs of 1 in 10 power 6, the probability is that unit in non-sterile in 1 in million. Already we discussed in terminal sterilizations. Okay, so the four pillars of robust aseptic processing, this is very important. What are the four basic pillars? What are the four basic points, all right, on which this aseptic processing is depending? I already we discussed. Person entering in the room should be trained and they should be monitored, because they can carry lot of microorganisms. Our hair can carry a lot of microorganisms, right? Our skin can carry a lot of things. Our dress can carry, our shoes can carry a lot of infectious substances. Even though everything, raw material, finished product, right, containers, closures, water fountain, everything was good, sterilized, but the person who is doing that particular work, person who are involved there, if they are not sterile, if they are not perfect, if they become the carriers for the pathogens, then no one can help them. So the first thing should be, first pillar of the aseptic processing, a person who is working over there should be technically sound, should be trained well in how to deal with these bacteria and viruses, right? He should know what will be the contamination, I should touch there or not, what should I wear, and everything, right? And that person should be monitored carefully. So we discussed these things in class 100, class 1000, class 10,000 rooms also, right? Who are allowed and how many people are allowed and how many people are not allowed. So this is very important. There are restricted rooms, restricted areas where everyone cannot allowed, so they should be monitored by access cards. Only authorized person can enter into the particular room, because other people cannot enter. So these all things should be monitored carefully, right? You are, even now you are seeing, especially in controlling and monitoring the corona, we are managing and controlling the people entering into the room and entering into the areas. You can see malls, airports, everywhere, right? Even schools and colleges. Why we are saying stand, you wearing mask or wear gloves, use the sanitizer, right? And all these things, headgear and everything, pp kids and everything, what this is, isolating a person from the viral burden and all those things, right? So first pillar is personal training and monitoring. Next, environmental monitoring, it's very important, right? Then you have, for environmental monitoring, you should have designed HEPA filters and laminar air flows. Then process stimulation, media fills, right? So process stimulate chassis, whether to check whether this particular area is sterilized or not, right? Like I said, you, if I want to check whether this particular working bench is sterile or not, I will swab it and I'll go into the culture medium and check whether it's sterile or. So always sterility checking should be done of the air present there, material present there, right? Containers present there, working bench, everything, right? So you should take media fills, and every process should be always validated and stimulated and checked whether it is sterile or not, right? Then these are four basic pillars of aseptic processing, right? Personal training, what kind of training should be given to the person, right? Avoiding contamination means knowing the potential source of contamination. First thing, how can we avoid the contamination? How can we avoid the spread of coronavirus? We should know the sources of contamination, right? We know that human fluids, right? Maybe sneezing, or you are spitting somewhere, or you're putting fingers in your nose or some ears and you're putting it somewhere else, it can spread the virus. So you should know the source. That's very, very important. Then only we can control it, right? So first thing, then personal equipments, air or liquid, drug products, containers or closures, outside environment. So from what is the source of contamination? So here person can be a source, yes. Equipment can be a source, yes. Air or the liquid like water or other vehicles you are using can be a source. Drug product can be a source. Container and closure can be a source, and outside environment can be a source. So we should keep them all in control. How person entering in the room, how he will be controlled? He should be trained properly. He should know that all these things are the sources of contamination. He should wear all the protective clothing. Equipment should be washed well before and after parental manufacturing and production, right? And they should be cleaned properly, they shouldn't be washed with them again water for injection or well heated and well sterilized and bacteriostatic water, not with the general water, because that general water will again can contaminate your equipments, closures and containers. Air entering in the room is controlled. How? Room like this, which have maybe some holes or windows open, is not suitable for the production. Everything should be closed, and the chance of air entering into the room should be only through HEPA filters and laminar airflow only. No window should be open, no opening, and no connection from the external environment is very very important, because outside air can be different. It can contain certain pathogens, it can contain certain contaminating products, it can contain dust products which can enter into the room. Even though if your parenteral is can be free from pyrogen, can be free from pathogen, but if any dust particle is also present, then your product batch will be rejected, because in particulate matter test we will see we don't want any undissolved dust particle also. So you should cut the connection from the outside environment. You should allow the air to be screened air, quality air, right? With proper humidity, with proper air quality, with proper speed, and free from all dust, bacteria, and everything should enter into the room. And especially environmental monitoring. In environmental monitoring, how I would sterilize this complete room. I have different ways. I can use UV lights to sterilize this complete room, right? Or I can use I can use gases like ozone, ethylene dioxide, ethylene oxide, right? And I can sterilize, fumigate this room and sterilize this room, sterile this room properly, right? I can clean all the working benches, floor, and everything with the disinfectant agents, right? I can spray the disinfectant agents. I can sterilize this environment and stop the communication from the other rooms. Then this room particularly will become aseptic room. Drug products entering into the room should be through exchange rooms only, carefully controlled, monitored, right? And sterilized. And containers and closures, as we said, not only equipment, filling equipment, but closures and containers should be also cleaned with sterile bacteriostatic water, right? Hot water, so that you can kill all those things. Yesterday we discussed gram negative bacteria will be there in water, and it should be killed only through autoclaving at 120 degrees centigrade at 15 lb pressure for 15 minutes, right? And outside environment communication, all these should be controlled and monitored. Anything brought in contact with or in vicinity of the product is a potential source of contamination. So if these all things, if they are coming in contact, right, and if they are not monitored properly, sterilized properly, that can be the reason for contamination. It may be a person, it may be an equipment, it may be an air, it may be a liquid, it may be a drug product, it may be a container, closure, or outside environment. If you're exposing yourself to any of these things, it can be a potential threat for contamination. Got it? Right? Type of contaminations, there are a lot of type of contaminations, viable and non-viable particles are there. This is not microbiology class, so you don't need it. Like I said, you right, the rooms should be like shouldn't be open, that broken windows which is allowing air, light, and everything inside the room. Such kind of room is not at all allowed, right? So sneezing produces hundreds of thousands of aerosol particles, right, that can then attach to the dust particles in the absence of any filtration system. And these particles, which may contain bacteria, spores, or virus, may be present in air for weeks together. If a corona patient sitting here sneezes, right, that small aerolized particles, right, coming out from your sneeze mixes with the dust particle, and it can be viable and it can stay there for 1 week, right? And some other person enters there in that room, that shop, that market, or that seminar hall, or that class, right? And he starts playing, and if he get contaminated, he touches like this, then again he puts his finger in your mouth or in your nose or your ear, directly the bacteria will go into his mouth, and it will can cause you the infection. So you should be careful, right? Then humans and bacteria, over 200 different species of bacteria are found associated with humans. Bacteria are found in intestines, in eyes, in mouth, in hair, in skin, in nose, and everywhere. Dry skin can have thousands of microbes per meter square, right? Millimeter square, right? So especially staphylococcus epidermis is commonly seen when your skin is scanned. right? So there is a lot of association with the human and bacteria. So not only bad bacteria, even good bacteria is there, and it is a part of the ecosystem, and for our survival even we need bacteria, right? So endotoxins, I hope you remember what are endotoxins. Those endotoxins which we are saying should be removed in water for injection, they are pyrogens. Pyrogens are not bacteria, they are not living, they are not viable. They are bacterial endotoxins, right? So endotoxins are very important. See how they're affecting macrophages. They'll attach the endotoxins and they will cause the tumor necrosis factor and intraleukins 1, 6, and 8. Right? And what are the spores and why are they major challenges? I think you already studied this in microbiology and we don't need it, right? But simply you can see this germination, right? Vegetative growth and spores, how the spores will grow, and simple spores which can stay, which can live for years together when they will get appropriate environment, proper humidity, proper food and everything. They will start growing and they can cause contamination. Viral contamination, I hope you should know this very well, right? Viral DNA can replicate, right? Transcription can occur, right? And it can attach to the proteins. Viruses are the small, the sizes in nanometers, the non-living entities that hijack the machinery of host cell, and they can cause the infection, right? Virology and knowledge on virology is very, very, very important. Sources of contamination, there are many sources of contamination, right? We need to control. So what you, what you let these people into your processing area, would you let these people in processing area, right? If they are talking with a lot of sputum, or if you're sneezing, what will happen? Complete destruction, right? So whatever you follow, that will be not only in processing areas. Nowadays we don't allow them in classes and schools and colleges also without any proper filtering equipment. That is your mask. If there is mask, then we are safe. That too N95 masks. If not, again all these particles will be spread, and now they are mixing it with the dust particles, and that dust particle can stay for weeks together, and that can infect you. So you should be careful. Sick people aren't the only people for contamination, and sometimes healthy people can also, right, bring contamination, and they can carry contamination. The skin is home to a virtual zoo of bacteria. Remember, your skin is a zoo for hundreds of types of bacteria, right? So see, person entering into the room in a septic area, how it should be monitored. I said you there will be an exchange room. Yes, there will be an exchange room with a lot of protective clothing. So if people are a major source of contamination, how do we avoid contamination? The product while we process it. So we will wear all these things. We will wear headgear, we will wear mask, we will wear, right? We will wear shoe covering, apron, and everything will be, right, from toe to bottom, everything closed, and our skin except eyes, nothing should be exposed out. Even for eyes, there will be protective glasses, right? So from our skin, from our hair, from our body, nothing should come in contact with maybe an equipment or closure or container or product or raw material or the air present in that room or the environment present in the room or the door or working table. Anything shouldn't come in contact with our skin, our hair, our body, like sputum and all those things, right? Then we will become the source of contamination. So we should be careful in that way, right? So first step is eliminating the source of contamination. So eliminating the source, we have studied all the sources, we have seen all the sources. So first step will be the eliminating the source of contamination. So well designed and maintained operated aseptic processing minimizes the personal intervention. People entering in the room should be restricted, minimized, and authorized person only should enter in the room, again with all those protective clothings, not barefooted, not with open head, not with big beards, not with all these things. Everything should be covered, right? So as an operator activist increases active as operation activities increases in aseptic processing operation, the risk to finished product also increases. If the people entering into the room increases, then risk to the final product also increases. So you should be carefully monitoring it. Second step is to reduce the risk of contamination through sterile barriers, aseptic techniques, and environmental monitoring. First, you should prevent the source of contamination. Second, to reduce the risk of contamination through sterile barriers, aseptic techniques, and environmental monitoring. Seriously, when you go to the industrial visit, maybe in 4-2 or something,